Radin. It's a pleasure for me to be the host of today's fine seminar. And like always, we have a quick look forward and backward. Last week, actually, I was myself the one presenting when I talked about harshness and the harshness response. And that, from my point of view, there's a difference if animals have to reduce energy expenditure to endure environments versus in the stress response, they increase energy expenditure to overcome stressors. And next week, there will be also a special, because Heike Rödel had to cancel a special panel discussion run by several students that are working together with Eduardo. They invited 4 editors or co-editors of journals that are publishing in our field. And the panel discussion will be about shifting analytical practices and institutional change. So we have a couple of students that come up with questions how these editors recommend how to deal with statistical problems and statistical issues. And that will be quite interesting, I think, especially for our young colleagues, but also for everyone else, because it's really going to the point and what to do with our data and when to trust it and what to trust it. But today it's a great pleasure for me to have as guest Lee Koren from Bar Ilan University in Israel. So this fine seminar is very nice that we can meet many colleagues that we think we know well, and we met 3-4 times in our life. Now we can meet them weekly. And Lee Koren is a colleague of mine who I know academically for many years, because I read so many of her papers that are of great interest for me, but which I never met in person. And so actually today is the first time we really met in person, and that's also a great advantage for me. Lee did all of her academic career in Israel. She did a master's thesis and PhD at the university in Tel Aviv, and working on social behavior in rock hyrax. She has been always interested in social behavior, and she was looking for a mammal that is social but also diurnal, active during the day, and can be easily observed, which in Israel is difficult because it's often long times of the year it's very hot there, and that's why most animals there are nocturnal. And with the rock hyrax, she found a great study system. And she herself started a rock hyrax project in the En-Gedi Nature Reserve in Israel, close to the Dead Sea, a very beautiful piece of earth with real wildlife. I've been there many years ago, several years ago. So if you ever go to Israel, I would recommend you to visit it. And so in 1998, she started this project as a master student, and also showing how important it is that we have some people in our field that bring in the enthusiasm, that invest the time, that invest the energy to start such new long-term projects, which is unfortunately very rare, because most students are normal humans and they prefer to sit in the comfort of the home in front of the computer. But I still believe we also need people going out there to the field and observe what's really happening. So Lee published way more than 60 papers, mainly on social behavior, also on vocalization. She did groundbreaking research in the fields of behavioral endocrinology, like analyzing hormone samples, hormone levels from hair samples non-invasively, a new technique that she helped to develop. And yeah, she, like I said, she did her PhD in Tel Aviv, and then 2009 to 2012, she went to Canada to Catherine Wynne Edwards to do a postdoc to further improve her knowledge in behavioral endocrinology before returning back to Israel, then to Bar Ilan University, which is also in Tel Aviv, 2.5 hours drive from there to her field site, and then 1 more hour hiking through the mountains. And she worked not only on rock hyrax, but also on gerbils, so one nocturnal mammal, on nutria, an invasive small mammal that we also have here in Europe. She is doing some studies also on human social behavior in Arabian bubblers, but I know her most from her work on the rock hyrax, and now I'm looking very much forward to hear about her talk on Mars and Venus, sexual conflict over testosterone in wildlife. And let's hope that the sharing now works.
Okay, let's try that. Do you see my screen?
Yes, looks good for me.
Awesome. So thank you, Karsten, and thank you everybody for the invitation. I've had a few hours now of meeting with some of you from the community, and it's been really a great afternoon. So I'm really thrilled to be here and share with you some of the things that I've been working on. I just realized today when I was showing my presentation to my students that some of the younger people in the crowd might not be familiar with a cultural reference. Men are from Mars and women are from Venus. So just so you know, it's a book that was written by American author and relationship counselor John Gray in 1992. And the book states that most common relationship problems between men and women are a result of a fundamental psychological difference between the sexes. So we're not going to get into relationships for humans, but I think the title fits, and you will see some of the data that I have concerning sexual conflicts between males and females in wildlife. So even when organisms may engage in a mutually beneficial interactions such as copulation, they bring to it a different set of costs and benefits. And although males and females share traits, their motivations and needs may be really different due to their life history disparities that lead to divergent selection pressures. Among the proximate mechanisms that are underlying the differences between the sexes are hormones that their job is to mediate the development and the activation of suits of traits. So today we'll mostly talk about testosterone. Testosterone is associated with morphological features, with physiological processes, and with social behaviors in both sexes. And although it's mostly studied in males, and that's because in men, there's about 7 times more circulating testosterone than in women, the relationship between testosterone and all kinds of features might be relevant for women and for females as well. So the relationship between testosterone and male copulation success, aggression, and also mate choice are really well established in males in multiple systems. And the trade-offs between parental behavior and reproductive success may also be testosterone mediated. However, even if testosterone is present in similar concentrations in the circulation, it often affects males and females really differently. For example, there are studies on dark-eyed juncos that have given us a lot of amazing insights through the work of the Ketterson lab over the past few decades through manipulation of circulating testosterone levels. And their studies showed that there are sex differences in the relationship between testosterone and mating related and all kinds of social behaviors. So today I want to share with you some of the insights that we gained from studying testosterone in both sexes and what it could tell us about sex differences and fitness related strategies. I will share accumulated evidence from 2 systems that we studied in my lab at Bar-Ilan University on how testosterone concentrations have different costs and benefits relating to the social status and copulation success, and that will be in the hierarch system, and also to growth and development when it starts really earlier on in utero, and that will be in the nutria system. And maybe we'll also get to mentioning cortisol and other steroids that affect testosterone levels. So the first system that I will talk about is the rock hierarchs, which are highly social. They live in groups that consist of both sexes. There are females and all their pups. There's 1 or maybe in some groups 2 resident males and some late dispersing males. And we've seen and used different metrics to show that in most groups, the females are the dominant sex. As you could see that there's limited sexual dimorphism. It's really hard to tell if you look at this group of furry mammals, who the females are and who the males are. I know who the male is, and that's just based on the individual marking that are found. I don't know if you could notice, this picture is taken in the winter when they have this amazing winter fur. So they're very furry, but you could see that they have collars around their necks that have their identity on it. And that way we could know who's males and who's females and who they are. In this system, the males are the ones that disperse. The females usually stay in their natal group, but they could move as well or break off from the main group. It really depends on the situation. Males could either be residents, that means that they're living with a female group, or they could be bachelors, and that could mean living alone, totally solitary, or living with other males. Residency is not related to the age of the hyrax. It's related to how well they sing. And that was part of my PhD. I'm not going to talk about vocalization today, but females choose males based on their singing, which portrays a lot of their features. And then the tenure of a male that is chosen is usually an average of about 3 years, but they could be with a group of females for up to 5 years. The bachelors, on the other hand, are local males. They associate with females of several groups, and they may attain very high social status. So it's not necessarily that all the dominants are residents and the bachelors are lower social status. It could go in many different ways. So this is a long-term project that I collaborate on with Eli Geffen from Tel Aviv University and Amiel Ilani from Bar Ilan University. And we've had many students throughout the years, master's students and PhD students, that have helped us collect the data that I will be sharing with you. We've been working on the rock hyrax for over 2 decades in the Engedi Nature Reserve. We capture hyrax annually. This is me during my PhD, capturing a hyrax. We individually mark them, and that allows us to observe them and observe their behavior. We look at the agonistic behaviors from which we calculate the social status using David's score. And we look at the affiliative behavior, and we use that to calculate social networks, and also on mating behavior, maternal behavior, and all the social behaviors that they engaged in. So earlier on, one of the first findings of this system is that in rock hyrax, the females have the same circulating testosterone levels as males year-round. We thought at first it was very unique to Engedi. We thought maybe it was unique to the hair testing that we do, but after testing many hyrax groups throughout the country and testing blood samples as well, we see that it is true. It looks like testosterone is the same levels as in males and females, and it's actually one of the highest levels that are known throughout the whole animal kingdom. So they're equal and they're very high, and despite that, the females reproduce every single year. The gestation period is very long. It's almost 8 months, which is very long for such a small mammal. And they nurse multiple offspring for up to a year. So maternal investment is definitely very high. Females produce between 1 and 6 pups annually, despite their high androgen levels, and then they communally care for them. In the past, we found that whereas males show a trend that we know from many different systems. The trend line for the males are the line, the broken line, and the trend line for the females are the solid line. On the x-axis, we've got the social status. Can you see my mouse? No, not sure if you can see my mouse.
Yes, we can see it.
Okay. So on the x-axis is the social status, where number 1 is the most dominant animal, and number 12 is the least dominant animal, and on the y-axis is the testosterone levels. So we found that whereas male testosterone is positively associated with their social status, with more dominant males possessing higher testosterone levels, in female, the association is totally inverse. So more dominant females possess actually lower testosterone levels than low-ranking females, suggesting that testosterone might be costly to females. Next, we wanted to test the relationship between testosterone and mating success. Since there are no sex differences in testosterone levels in hyraxes, and they're considered high, as I've told you, our predictions were mixed. On one hand, we expected testosterone to be related to copulation success in an opposite manner in the two sexes, like we saw for social status. On the other hand, we suspected that considering hyrax circulating levels, male and female copulation success will show similar trend. So we observed 494 mating-related events. This is over 12 years, which included 6 different social groups. The mating period in the hierarchies is really short. We're talking about just a couple of days a year, and we of course don't know when they are. So we're observing them throughout the hottest time of the year in July and August until that one day comes about and all of them mate. Both sexes mate with multiple partners. As you could see here, what you see here is the sexual networks of 2 different years. We've got the 2011 and the 2012 sexual networks. The arrows show who initiated the interaction, and the width of the arrow is related to the number of sexual interactions that we observed. And the red, the females are in red and the males are in blue, where the bachelor males are the sort of diamond shaped and the resident males are the triangles. So you could see that some males mate with multiple females, some females with multiple males, and then there's some pairs that are pretty exclusive. Overall, we found that females are more choosy. They tend to reject more copulation attempts than males. We think possibly females do, though, mate with multiple males, possibly to confuse paternity or to allow for sperm competition. We've seen a few times that males attack pups, and there's also a single direct observation of an infanticide that has been documented. So females do have a reason to maybe confuse paternity. Both residents and bachelor males have similar copulation success, but residents tend to mate guard more, specifically the mate guard females that were mothers in previous years. We expected choosiness to be related to testosterone in both sexes, and that higher testosterone males and females will mate more and be less choosy. This is something we know from humans and also from the dark-eyed juncos, that individuals that have higher testosterone tend to be less choosy and mate with multiple partners. In humans, actually, men also that have higher testosterone tend to cheat more and have more partners. So it's something that is across systems very well known. We also expected that males with high testosterone will mate more, just like in humans, and mate guard less because they will be busy mating with other individuals. So first, we wanted to see if it's possible if there might be something about sperm competition, and thinking about how males differ in their status. We're currently working on sperm competition and how that may play out in different male strategies, as residents do not have a copulation advantage over bachelors, but maybe their sperm quality or quantity differs. So we have a protocol to collect sperm in the field. It's a very difficult thing to do. This protocol was validated by undergrad vet students in the lab, along with our collaborator, Dr. Tal Raz. He's from the Hebrew University, and my master's student, Hunter Warwick, that is now collecting sperm in the field. And you could see them here in the picture, the two of them. So we found some really amazing basic biological facts looking at the sperm. For example, the hierarch sperms are the only ones. This is the picture on the right. Hopefully you could see it. Around their head, they have a little spurt. Tal, who's worked with many species collecting sperm, he says was very excited. It's the only species he knows that have the little skirts in them, and also have other features that are very unique only to hierarchs. Actually, almost everything that we find about hierarchs seems to be unique and amazing and exciting, and keeps us working all this long term in the field. So sperm is collected in the field. It's stained to detect normal. These are the white. We're looking now in B. So the white ones with a white head are the normal sperms, and the one with a pink head are the dead sperm. We also look at spermatozoa, which are damaged. That's the bottom picture. We also look at testicular damage. You can see here, the arrows are pointing at a few cells that show testicular damage as a sign of stress. So these are really preliminary findings from the first and second year of this 3-year study collecting sperm in the field. So we found that although bachelor and resident males copulate at the same rate, it seems to be differences in sperm characteristics between the males that reside with females and the bachelor males. So we've got here on the left, the bachelor males and the resident males. In the blue are the samples that we took in July, just before the mating period. And in the red are the samples that we took in August, just after the mating period. So we could see that there is a decline in sperm after the mating period, but only in resident males. We still don't know what that means. Does it mean that they use more sperm? Does it mean that they get sperm depleted? Many questions that we'll be looking into in the next few years. Okay, I'm stuck. Okay. So back to testosterone and mating. We used a data set that included 69 male samples. This is Ishai Weissman, a PhD student that worked on testosterone and vocalization in male hyraxes, and in Bar Schnitzel that looked at testosterone and maternal behavior in female hyraxes. And so we had hair testosterone values for all of these individuals. These allowed us to have 86 male to female combination where we knew the testosterone for both partners in their interaction. Overall, we had 199 mating related events for these individuals, 140 for the, of bachelor males and 59 of resident males. And for the analysis of copulation success, we used 335 events in which either copulation or rejection were observed. And here I'm showing you the 129 of them that we had testosterone levels for both males and female partners. So in all the graphs I'll show you afterwards, the male testosterone is on the y-axis and the female testosterone is on the x-axis. And you see the circles, some of them are overlaying many individuals. The ones that are white are ended up in rejection, and the ones that are full circles ended up in copulation. Go. Okay. So we detected a significant effect of both male and female testosterone on copulation success. What I'm showing you is the fully factorial model. Again, we have the y-axis shows the male testosterone, the x-axis shows the female testosterone, and the colors denote the probability of copulation success, where warmer colors such as red show a high probability of copulation success, and a cold color such as blue shows a low probability of copulation success. So the model revealed that the interaction between testosterone levels in males and females was significant. And you could see here the red corner. So whereas copulation success increased with the rise of testosterone in males, it decreased in females. In other words, females with the lowest testosterone levels and males with the highest testosterone levels are the ones that had the highest copulation success. Whereas females and males with low testosterone level seem to have lower copulation success. We did not find an association between testosterone and choosiness in either sex, and we did not find testosterone levels to be involved in the initiation of sexual interactions in either sex. So both males and females were found to be in both roles as initiator and as receiver in different events, and testosterone levels between the two roles did not differ. And testosterone was also not related to rejection rates in either sex. So to summarize just the Hyrax findings that I've showed you until now, we found that there is a disassortative mating in respect to testosterone. And we think we have some clues to the cost of testosterone for females in terms of both social status we saw earlier on and also population success. And I think these results open up a lot of intriguing questions relating to the role of testosterone in mediating similar trade-offs in males and in female reproductive success. High female testosterone, the hierarchs, could be a byproduct of high male testosterone. Maybe they co-evolved as a consequence of selection on males for increased levels. And since selection differs between the sexes, a trait that is beneficial for one sex may be detrimental for the other sex. And similarly, if male and female testosterone are correlated, then an elevation of male testosterone may be constrained by females. Intersexual genetic correlation may also be the origin of the negative effects that are associated with high female testosterone levels. And that brings us to the second project that I want to share with you that looked at how early sexual conflict starts. So we hypothesized that it might start in utero really, really earlier on during embryonic development, and that these effects may be carried on into adulthood. And for that, we turn to the nutria, which is a really successful and invasive species worldwide, which leads to being culled in many countries, including in Israel. So this study is also going to be long-term. Right now it's 10 years and running. It's in collaboration with Yoni Volkman. He's from Tel Chai College in Israel, and Uri Shines and Rachel Ben Shlomo Ekli from Haifa University. And the students that worked on it was now Dr. Ruthie Fishman. She did her master's and her PhD in my lab on this system. Ariel Yael, who did her master's, and now Noi Bendo, who's doing her master's and hopefully will stay on. So why did we go that far? If we look at fetal development, sexual differentiation, and also maturation, they could all be affected by the sex of the neighboring fetuses. If they create a local hormonal environment that might drive individual differences. Testosterone can transfer between fetuses, affect not only the fetal development, but also the life history and the fitness long-term. This effect is called the interuterine effect, and it's been documented in many species and has been associated with individual differences in morphology and growth rate, in behavior, and also reproductive ability, and they all carry on into adulthood. For example, mating behavior, both lordosis and also mounting behavior, as well as how attractive the individual is perceived by the opposite sex, is known to be affected by the intrauterine effects. For example, in mice, it's been really well researched that females that are located between two males have higher testosterone levels than females that are not located between males. And they also have difference in anatomy and physiology and reproductive ability. And males, for some reason, prefer not to mate with them. They prefer to mate with females that were not next to males in utero. The interuterine effect is also associated with aggressiveness and home ranges, also with scent marking, with parental behavior, and also with the number of offspring and their sex ratio. So if we're talking about mating behavior, aggression, territoriality, what may mediate all of them? Right, testosterone. So that brings us to talk about testosterone again. But before that, I just want to talk about the model that we use. The nutria is a semi-aquatic rodent that is native to South America. It has really large litters and a very long gestation period relative to its weight. So the fetuses have to cope with the hormonal pressures that their neighbors exert for several months. Nutrias have some of the lowest recorded testosterone levels in nature, opposite to the hierarchs. Remember, I told you that has the highest. And we receive pregnant nutrias from culling efforts in the Hula Valley in Israel and study the embryos while they're inside the utero. So here we see the utero, the nutria uterus. You can see the male fetuses are circled in blue and the female fetuses are circled in pink. And in order to investigate the effect of the interuterine position, we have to devise a scheme that describes the location of the uterus relative to the other fetuses in the utero. So since in most mammals, male fetuses have higher testosterone, it has been shown that testosterone could be transferred between fetuses through the blood and the amniotic fluid. We could describe the location of the fetus in proximity to a male fetus. So that notion has been proposed by von Saal many years ago and been really well used. So 0M are individuals that don't have any males next to them. For example, this male over here, the blue male, doesn't have any other males next to it, so that is 0M. On the other hand, the females here, that is in pink, she's got males on both sides of her, so she's 2M, while this male over here has only one male next to him and a female on the other side, so he's 1M. And that's one scheme that's been really useful. For other species, other schemes have been found to be more useful. For example, in rats, the location downstream to males has been found to be really useful. In pigs, it's the proportion of males in the litter. And another possible method, which Ruthie proposed, is the proximity to the opposite sex, in which a fetus is proximate to the opposite sex. It's denoted P1. Some of these fetuses have a fetus of an opposite sex next to it. And if it has only fetuses of its own sex next to it, we call it P0. So all these females in the uteron, in the horn here are all P0. The other thing we had to look is the anogenital distance, or the AGD. And that's been known to be an indication of exposure to testosterone during fetal development. In the nutria, just like in other mammals, females have significantly shorter AGD than males, and we use that to sex them. I mean, before 80 days, we use internal anatomy. You could see here the male testes and the female uterus. And here throughout the different ages, you could see in the females. We could later on use then the anogenital distance. From about 80 days on, you could see here that in females, they're very close together, and in males, they're farther apart. In mice and rats and in gerbils, the length of the AGD is associated with a fetus's proximity to male fetuses. And Ruthie found also the same thing in the nutria as seen in all the other model rodent. So that both males and females that were next to a male in utero tended to have longer AGDs. So that suggests that there is a masculinization effect of the proximity to a male in utero. So first, Ruthie wanted to look at the mums. She looked at the testosterone of the mother and found that females that were heavier had male biased litters. So this is the graph that we have here. On the x-axis, we've got the maternal weight. And on the y-axis for all of them, we have the sex ratio, where where 1 is an all male litter and 0 is an all female litter. So you could see here that females that weigh more tend to have more males in their litters. One of the first hints that Ruthie found that high testosterone might be challenges to females is that females with higher testosterone levels, right here on the left graph right now. So the females with higher testosterone tend to have lower sex ratios, meaning they have litters that have more females in them. What we're seeing here are the estimates from the models. Males seem to be costlier to produce. Usually the heaviest pup in the litter, in most cases, is males. And males also suffer higher mortality rates in utero. This is how they look. See right here, there's a litter where there's 2 shriveled pups. I circled them in blue. You could see that they're shriveled, they're growth stunted. And we also found that the older the pregnancies, this is now the middle graph, you could see this is the pregnancy age in days, and the older the pregnancy is, the lower the sex ratios. So it seems that male fetuses are being aborted or being dissolved while in utero. Next, Ruthie looked at the fetal testosterone. So now we're looking at the fetuses. This is a picture of a 140-day-old fetus. You can see that it's fully covered with hair. It also has nails, it has teeth. It's really ready to be born. So overall, she found this is the male fetuses and this is the female fetuses. Overall, she found that males had higher testosterone levels than females. But when we look at the intrauterine position, and here on the x-axis, we've got the P0, the males that were not next to females, the P1, the males that were next to females, and also for the females, that were that were only next to females and the females that were next to males as well. So when we looked at that IUP, we discovered that females that are not proximal to males, these are these P0 females, they have testosterone levels that are not different from those of males. The letters denote the statistical differences between the groups. Oops. So you can see that these females had this not different testosterone levels than males. And surprisingly, it was very surprising because remember when I was talking about mice, we thought that the females that would be next to males are the ones that would have higher testosterone. We found that actually the females that were not next to males, they're the ones that had the lower testosterone. So Ruthie thinks that it's possible that it's the length of the pregnancy that is very important here. The only other species with very long gestation that was studied was domestic pigs. And in them also, there was no correlation between the proximity to males and testosterone levels in the very late progressive stages of gestation. But the length of the AGD, that is fixed much earlier stages of gestation, was correlated with the presence of males, just like we saw in the nutrias. Testosterone has been shown to have an adverse effect on reproductive ability in females in many different systems. Also in humans, women that have polycystic ovaries have higher testosterone and more issues that are connected to reproduction. And these results that we see imply that while at the earlier stages of gestation, nutria females are affected by their proximity to males, and that we see that by the length of the AGD, there might be some kind of mechanism, for example, developing lower sensitivity to testosterone, maybe lower production of endogenous testosterone, or a more rapid rate of testosterone clearance that helps females avoid the adverse effect of masculinization by testosterone in such a long pregnancy. So we do have evidence that there's possible sexual conflicts in utero, males and females adversely affecting each other. There's sex differences in testosterone levels. In the hierarchs, there was no differences in testosterone levels, but there were sex different associations, and we thought that sexual conflict arose from similar testosterone levels and possibly intersexual correlation. But in the nutria, we see that they have differences in testosterone and still are not sure if that is attributing to sexual conflicts. In other species, such as in red deer, we know that there is a within and across genetic correlation. In red deer, the males that have higher testosterone also have higher reproductive success and also higher testosterone in their offspring, which is great for their sons' fitness, but is detrimental to their daughters. In the nutria, we found multiple paternity and possibility for heritable component of the fetus, of the testosterone in the fetuses, because we found that the sons of the most prominent father, that has the biggest share of paternity, had the highest testosterone levels, and those that were sons of rare fathers had lower testosterone levels. And we did not find such a relationship in females. So we suspected that it's possible that in nutrias, at least, there is heritability only within the sex. And we wanted to check that in the nutria. First of all, whether testosterone was heritable and whether there is sex assortative heritability. So this is just preliminary data. It's a sample that includes 91 fetuses. We have 44 males and 47 females belonging to 16 different litters. And all of them are in the last trimester of pregnancy, so that they already have hair and we could measure their testosterone in the hair. All of them were genotype and also analyzed for siblicity. And then we used the REML model. We included maternal ID and paternal assigned ID as random factors, and also maternal testosterone levels. And we found really different contributions of the maternal and the paternal factors to female and male's testosterone levels. This is the female, and this is the male testosterone levels. So in the female fetuses, the maternal factor is responsible for 49.4 almost percent of the total variance between the female fetuses, while the paternal factor is responsible for only 22.3% of the variance. And in male fetuses, we found exactly the opposite. We found that the maternal factor is responsible for only 5.6% of the total variance that was observed, while the paternal factor is responsible for 57.6% of the variance. Using the animal model approach, we also estimated heritability between the mothers and daughters, and mothers and sons, fathers and sons, and fathers and daughters. And we found that the testosterone, this is the heritability estimates between the parent and offspring of the same sex, were much higher than the heritability of the cross sex of the opposite parents, offspring of the opposite sex. And the heritability of the testosterone from fathers to son and mothers to daughter was much higher than the cross sex. We also found that the estimate of the cross-sex genetic correlation was about 0.6, which suggests that there is a certain degree of genetic independence between the sexes, and that there is a sex assortative heritability, at least for testosterone. So this is where we stand now. But I want to mention that testosterone is not the whole story. I mean, measuring testosterone is all nice and interesting, and I've been doing that for many, many years. But more and more, we realize that it's not the concentration of testosterone that is important. It's what translates into effects is only after it binds to receptors in the brain that translate into changes downstream. And we've been broadening now the questions about heritability to sex differences across the HPG axis. So just for those who don't remember, the HPG axis, these are fetal brains from the nutria and the HPG axis. So the hypothalamus communicates through GnRH, which binds to the GnRH receptor, which is in the pituitary, which releases LH and FSH to the gonads, which release testosterone. And testosterone binds to the aromatase receptor, while it could also change via aromatase to estradiol and bind to the estrogen receptors, alpha and beta. So we wanted to look at as many possible components of the HPG axis to see whether there are sex differences in the different components. We've seen that it is present in testosterone, but to see whether in other components it is present. So we looked at the mRNA levels for the different genes that code for the receptors and also for the enzymes that are related to the axis. We also looked at the GnRH and GnRH receptor. We've got here the androgen receptor, aromatase, and the estrogen receptor alpha. And for all of these, we've got here the males in the blue and the females in the red. I know it's really small. I just wanted you to have the general effect that there is higher female expression levels across the board. And I circled the significant differences that we find so far. This is for a sample size that has 28 males and 21 females. So it's just initial findings, but it already shows that there's something interesting going on, and there's differences in the brains and in the way that testosterone and other steroids are communicated in the different sexes as a function of who are their neighbors in utero. There are differences between those that are next to their opposite sex or those that are only next to the ones to their sex. So on the y-axis here, we've got the relative expression levels. They're all normalized this is real-time PCR data. They're all normalized for the expression of the housekeeping gene, and we use the GIS1. And we could see that in different areas of the brain. So this is the hypothalamus, this is the cortex, and this is the striatum. but we have the other areas of the brain that we're still working on. I just wanted to show you and see where we're going in that. It's very initial discovery stage. The other interesting pattern that Riel found is that if we look at the relationships between different parts of the HPG axis, so here you've got, I don't want you to actually read it, but just get a sense that there are different genes in different brain regions. These are females and these are males. This is all in the first half of the pregnancy, so very earlier on. And we can see that there's a very strong relationship. These are only the significant relationship between the different genes. So we could see that in the females, there's a greater relationship than we see in males. We're not sure if that means more coordination in females is needed. Very interestingly, the androgen receptor here in the blue, that seems to be related in a negative manner to almost all other genes in brain regions. And we see that later on in pregnancy, this is in the second half of pregnancy, these patterns break down, meaning that there's much less coordination. It's possible that in the first half of pregnancy, during organogenesis, when the fetus is developing, steroids are much more crucial than in the final growth phase, when they're less needed, or at least the coordination is less needed. And we still need to pursue it and look more in depth. So until now, I spoke about sexual conflict, but I also wanted, just before I end, to show you some of the benefits of sharing the uterus for the two sexes. So here we've got results from that were published a few years ago for cortisol in utero. This is for 82 different litters of fetuses, and it's actually the same picture. This is the average cortisol. These are the average of the same litters. That here are the individual fetuses. In red are the females, and the blue are the males. And here we've got the sex ratio this time is on the x-axis, where 0 again is all female litters and 1 is all male litters. You could see that it seems like there's some kind of stabilizing selection where the fetus, the litters that have mixed sex litters seem to have the highest cortisol levels. So at first we thought that this is another piece of evidence for sexual conflict, because it looked to us that litters that are all females or all males seem to be happier. They have lower cortisol. They might be better off than litters that are more upset. They have higher cortisol, it's the stress hormone, and those are the mixed sex litters. But in fact, in utero, cortisol, especially in the last trimester, and I remind you that we're quantifying these in hair. If you guys want to hear more about it later on in the question period, if you ask me about it, I could talk more about how we quantify steroids. But hair is only found in the last trimester of pregnancy. So this is really at the end of pregnancy. And we found that in that very, very last piece of time of pregnancy, cortisol is crucial for lung development and also multiple other systems, the maturation processes of all the systems and preparing for fetuses for life outside the womb. And it seems like in those mixed sex litters that had the highest cortisol, the fetuses were actually the most prepared for life outside the womb. They were also bigger, they were more developed, and seemed to have an advantage. So evolutionary speaking, that could be keeping mixed sex litters in business. So to summarize this talk, we found sex differences in wildlife steroids and in the relationship between steroids and fitness-related traits, that it could suggest sexual conflict. And we saw that there are fitness costs that are reflected through social status, mating success. I don't have time to show you, but we also have data from wild gerbils in the field that show that there's different strategies in risk-taking behavior between males and females. We also work on the Arabian babblers and see that there's mixed strategies in cortisol, corticosterone, and feather corticosterone and dominance in that system. So it seems to be a line of research that is worth pursuing and will give us a lot of indication of what's really going on between the sexes. The next step is to assess how sexual conflict is resolved or maybe played out, whether it's heritable, and then follow them throughout development as a function of life history and also interactions with the environment. And with this, I'd like to end. And thank you again for inviting me. It's been a really great day and chatting with all of you, and I'm looking forward to your chatting further. And also reach out to you guys in the community if you know any people that are looking for master's or a PhD. We have open position for students researching hierarchs in the field, nutrias in the field and in the lab, and also in Arabian babblers. And I would love to take any questions.
Yes, thank you very much. Lee, really fascinating talk, and it's great to see these data on these long-term studies and how you then manage to combine this from the hierarchies with the nutria. I mean, it's we all know this is a huge investment of time and energy to run 2 projects like that. So if anybody has questions, we do it like always. Please type a question mark into the chat. And if you have one on YouTube, you can write it in the YouTube chat and I will have a look on it. And then if I call you up, then please quickly introduce yourself, say what's your name, where you're coming from, what you're studying, and then ask your question. And we will start with a question by Eduardo.
Thank you. I'm Eduardo Fernandez Duque, currently in Argentina from Yale University in the US. Thank you, Lee, for a very, very interesting talk covering all kinds of levels of analysis. So 2 talks are very, very interesting. I have a very practical question. Like you mentioned, we call them coipos in Argentina. And they're a huge problem in terms of exotic species. How do you get to Israel? Do you know anything about the kind of environmental problems they are causing where they are?
Yeah, so they've been brought to Israel in the 1950s for the fur trade. They have really soft fur, and they were brought by the hundreds, and they reproduce very quickly. Their pregnancy is about 140 days old, but once they're born, within, I think it was about 100 days, the females could become pregnant, and they have huge litters of up to 15 off fetuses and grow very quickly. By the 1960s, fur hats and coats became less popular. It wasn't fashionable anymore, and there were no benefits in keeping these animals, and they were just released everywhere. So they are now in all the rivers in Israel, and they have a vast effect on agriculture. They burrow in the riverbanks, and they eat the vegetation, and the farmers really hate them. And that's the situation worldwide. And that's why they're called by the authorities. And when I found out about it, they're called by the thousands. So every year they kill a lot. And so we just find out what days that they're calling them, and we just follow the hunters and collect the bodies. So we're very much against it. We're actually we're collaborating on a few studies how to manage their populations using non-invasive methods, using oral contraceptives, and etc. So we're against the culling, but they're doing it anyway. So we're doing some science with it, and it's been fascinating.
Thank you very much.
And I have some question. There's somebody coming up now. Wait a moment. Okay, then Claudia Fichtel has a question.
Yeah, hello. My name is Claudia Fichtel, German Primate Center, and I'm interested in the evolution of social behavior. Hi, Lee.
Hi,
Really
Claire.
nice talk.
Okay.
So I actually wonder, how do you collect sperm in the field?
Do you really want to know?
Yes.
It's not for the faint-hearted. We use electroejaculation methodology.
And you capture them.
Yeah, we have to capture
Okay.
them, state them, all with permits, of course, and then use an electroejaculator and collect the sperm. We have to do all the staining and also all the counting in this in the field. So we have a microscope that we carry into the field, and it's very intensive work. I've got an excellent master student who's doing it. This is his last year doing it. And we've collected quite a few samples, but it's been fascinating.
Well, thank you.
Welcome.
Then Suleima.
Yes, hi. Suleima Tang Martinez, retired from the University of Missouri in St. Louis. And I have a bunch of questions. I'm only going to ask two right now, and then if there's time, maybe I can get to the other two. But thank you very much. It was an absolutely fascinating lecture. One question that I have is, you talked about the costs of high testosterone in the females. I'm wondering if there are any benefits. I think, if I'm not mistaken, you mentioned that the females are dominant. And so I wonder whether high testosterone affects that dominance, and there's a reproductive success benefit to being dominant, or any other, any other benefits of being dominant. So that's, I'm sorry, any other benefits of having such high testosterone levels. And then the second question is, you mentioned that the hyraxes have the highest levels of testosterone for females. And I'm wondering if you know how that compares to the spotted hyenas, and any comparisons that could be made between the hyraxes and the spotted hyenas. And I'll save other questions for later.
Okay, great questions. First of all, for the females, we're actually, we haven't found any advantages to high testosterone in the hyraxes. We actually found that females with high testosterone are subordinate to females with low testosterone. So the most dominant females actually have the lowest testosterone levels. So we think that that's a major cost. And also with copulation, with regards to copulation, we see that the females with high testosterone have the lowest copulation rates. So we don't really see an advantage at this point. They seem to overcome it. They do mate every year with lower ranking males, but they still mate, and they do have offspring every year. We think that maybe the quality of the offspring may be lower, and this is the advantage of a long-term study system, that over many years we'll be able to assess whether females that have high testosterone have lower ranking or less productive offspring. Because they live for so long, they could live for up to 12 years. We have about 4 or 5 generations at this point, and we haven't been able to detect that cost. But I do think that overall there is a trend, and I think we will be able to see and be able to assess the effect of testosterone long-term for that system. You asked about the spotted hyenas, which are fascinating. So although spotted hyena females are masculinized, the adults don't have higher testosterone. The adult females do not have higher testosterone than adult males. When they're in utero, they have higher testosterone. They do have higher androstenedione levels, which is a less potent androgen It binds less strongly to the androgen receptor. It might still be important, but their brains are not as masculinized, and testosterone does not seem to be the main hormone that is driving their masculinization. With the Hyrax, we haven't looked at other androgens so far. I mean, we are planning on looking at 5-alpha dehydrotestosterone and also DHEA and androstenedione. It's in the making. It's being validated as we speak, and we hope to have a full panel of steroids that will be able to look at the other steroids that might be crucial in mediating all of these behaviors, and that might give us additional insights that we're missing right now.
Thanks.
Okay, then Jingyu has a question.
Yeah, hello, I'm Jingyu from the University of Strasbourg. My question is, how many sample size do you have when you study the heterogeneous, when you study the, sorry, I just suddenly forgot the word, is the
Hyrax?
not the Hyrax, but the inheritability, sorry, heritability.
For the heritability estimates, yeah.
So the preliminary results that I showed you had 90 fetuses. About half were females and half were males. I think it was 40 females and 47 males for that.
And they're all like related from the mother and daughter, father and the son.
There are 19 different litters, so they had the same, like each litter had the same mother, but we found that there is multiple paternity. So some litters had just one father, some litters had 2 or 3 or 4, up to 5 different fathers in the same litter. So it really depended on the litter.
Okay, for sample size of less than 100, these heritability estimates are quite strong and impressive. Then we have Maren Hook has a question.
Hello, my name is Maren Hook from the University of Derby. During my PhD, I've dabbled a little bit in hormones, but I don't really know much about it. How have other Afrotheria been checked for testosterone levels in males and females, and also for the sperm peculiarities? So whether there's a taxonomic thing there, but also in general, for how many species have the females at all been tested for testosterone? Because when I did my PhD and we, just for fun, we checked the testosterone values of our female mustache tamarins, and they were quite high, but we used fecal samples, so we didn't really trust our results, and it wasn't validated, so we never did anything with it. But we always wondered whether that is a genuine thing or not. But we didn't find much information about testosterone levels in females in general. So I'm not sure whether there might be actually more surprises out there if we look closer.
Totally, yes. I'm sure until about 20 years ago, since steroids were defined in the 1950s, I think, it was initially realized that men had much higher testosterone than women, and therefore, for all study systems and all animals, testosterone was only measured in males, and estrogens were only measured in females. And the reason why I measured it in both was just because I was a rookie and I didn't know that. So I measured it in both species, in both sexes, and found that they had equal levels. And then I went back to I worked the opposite way. I went back to the literature and actually found that there were no studies that measured testosterone in females. There were very, very few. Some in geese and in hyenas, of course, and in a few primates. But I think there were 4 or 5, just a handful of studies that even measured testosterone in females. And in all of them, males had higher testosterone levels. Having said that, since then, I've worked on over 30 different species of mammals and birds. And in almost all of them, males do have higher testosterone levels. I'm talking about bears and wolves and also the striped hyenas and many different species. And males do usually have higher testosterone, but it's not across the board. In gerbils, for example, males and females have similar testosterone levels. In hyrax, we've seen also in the Arabian babblers, males and female testosterone levels are similar almost year round. So it's not something that could be generalized automatically. And I totally agree that it needs to be studied per system and also per context to see if it's relevant and what in how it is relevant. About your older studies, I think the advantage of taking fecal samples is, of course, that they're totally non-invasive. You don't have to capture and collect samples, but when you study fecal steroids, you're actually studying the metabolites of the steroids and not the steroids themselves. So it's the steroids that were metabolized through the liver and the kidneys and excreted. And of course, they have to be really well validated in order to trust them. So yeah, still a lot of work needs to be done. Although I've been working for many years on steroids, I still feel like a rookie. You know, everything we know about steroids is sort of like still in a black box, and all of them affect everything, and they're affected by the context and the system. And I mean, that's what makes it so interesting, but also so complex and hard to make clear conclusions about their effects.
Okay, thank you. And a question by Kaya Tombak.
Hi, I'm a postdoc at Hunter College. Thank you very much for that talk. Just foreshadowing a little bit of what we want to discuss next time, I will have a statistical question or an analytical question. You have lots of important findings, and some of them show things we might not expect, and some of them show unclear results where there's a lot of variation, because hormone data are messy often anyway. But what have you developed as strategies for publishing the results, even if they're not clean, or even if they're not clean as in like very nice clear lines, and even if they're not what we expect? Do you have certain journals that you go to? Do you try to make the variation itself interesting? Do you have lots of data that are messy just in your hard drive that haven't gotten out? What have you developed as strategies there?
Yes, for all. I think, I mean, nowadays when data is shared, it makes it much easier because everything is, you know, much more open and easily accessible. But I think the whole, you know, publishing only relative strong positive association is unfortunate, because a lot of the interesting things are in the variation. So we do talk about variation. A lot of times that is the important and the interesting factor. And try to, I mean, our home journal is Hormone and Behavior. I mean, we also publish, of course, in other journals, but I think it's Hormones and Behavior. I mean, they're very open to publishing about many different systems and things that are not necessarily lab-based model species that are showing, you know, like very clean results, but they appreciate the diversity and talking about, you know, the noisiness and the dirty sort of data, and trying to work through it and the story behind it, and looking at the variance. And a lot of stories are not out yet. I mean, we're working because, you know, steroids are so, the variability is so huge. A lot of times we wait and we collect, you know, we work with big data sets, and that's another advantage of a long-term study, is that we could accumulate data across years and then have the story slowly come out. So I've told you guys before, I could see trends of certain things starting to emerge, but it's not there yet. Like we still need more time, we need more data to be able to see whether the story is there. And you need a lot of patience, but I think it's worth it. It also makes it ecologically valid, and once it's there already, then it's very, very strong, despite all the noisiness and the variance.
Good. Then I have some question to better understand your hormone data. When you maybe you said in your talk and I did not pay enough attention. When you say in rock hyrax the females have higher testosterone than in any other species so far studied, how high are the blood levels of testosterone in female hyrax?
I don't remember offhand. It's the titers are a couple of order of magnitudes above any other species. I could pull out the paper after.
Okay, so it's something like I mean, I know in striped mice, for example, it's something like 2 nanogram per milliliter, but then in hyrax it would be rather something like 20 or 40 or something like that.
Yeah, I'll send you. I'm writing down for me to send you.
For corticosterone, well, of course, there's much more data from more species. There's also a variation that, like striped mice, have 100 times higher corticosterone levels than most other mammals, but there's a few other mammals with very high corticosterone levels, like prairivoles, and one species of tamarines where it's 20, 50, 100 times higher. So this is quite surprising. But if I understood right, but maybe correct me, the data you showed in your talk for rock hyrax typically is from hair, is it?
Yes, it's all from hair. I've got one paper that is sort of a validation paper, the first one that reported the testosterone data, and I was so insecure about it that we went to collect blood and see that there is a relationship between blood and hair testosterone. But then from the nutria, it's probably from blood because it's easier. No, it's all hair as well.
Okay, but
yeah, blood's steroid levels is very challenging, because what is reflected in the blood is only like a picture of what's happening right now. Like it you know, you've got a wild animal that is captured, is very, very stressed in your hand, or is dead. So it's very stressed because it just died. And I think what you get from blood is not really related to anything in their life history. In addition, steroids in the blood are linked to carrier proteins, and they're not active. I mean, some of them are active and some of them are not, but what you get in the blood is the total steroid levels, the active and the inactive. The hair, on the other hand, is a long-term integrated measure over the time of growth. So you have the blood, the little blood vessel that feeds the hair as it grows, and over the weeks or month, depending on the species. For example, in humans, the growth rate is about a centimeter a month. So you could actually, I've got a couple of years of history here, and you could segment my hair into 1 centimeter pieces and look into 1 month resolutions. So if you're looking at long-term social status and reproductive status and so on, I think hair that is representing the long-term data is much more reliable. It also, hair only includes the steroids that are active, that are in the free state, because the proteins are too big to infiltrate into the hair. So they're carried into the bloodstream, but they're not carried into the hair. So hair is actually a more reliable measure for free steroids that are bioavailable and related to behavior and the context. So
how has this been shown that it's only the active hormone that's in the hair and not the one that's bound?
I think through radioactively measuring, radioactively marking different proteins. This has all been used. This has all been validated through forensic medicine. The Olympic Committee used hair testing, and also forensic medicine uses hair testing for all kinds of drug detection and steroids, and actually everything that's in the blood gets dumped in the hair. So I could take a hair sample from you and know if you smoked, or if you drank alcohol, or if somebody around you smoked, or any medication you take. Everything that is small enough gets deposited in the hair. And we have a history that we could take many, many years later. We have measured testosterone in hair from mammoths that are 70,000 years old, and from people that were buried thousands of years ago. So the steroids are cholesterol, they're derived from cholesterol. They're very stable, and they just sit there in the hair and they wait. So I think it's a possibility to show it. And in the nutria, we've got an animal that's already dead. The fetuses have hair that we know was grown during the last trimester of pregnancy. So taking the hair from the fetuses actually gives us indication of what these fetuses experience throughout the whole last trimester of pregnancy. So it's a long-term integrated measure. It's less good if you want to ask about how an animal is feeling right now when a predator arrived, or something very that is immediate. Then of course blood would be preferable, or saliva. Saliva also only has free steroids in it. Saliva does not have the proteins, the carrier proteins. So saliva is better than blood in that respect, but it shows the same sort of time period of just that moment. As I said, fecal and urine hormones are great because they're non-invasive and they open the window of analysis, but they're metabolites, they're not steroids themselves. And hair and nails and all kinds of keratin matrices such as feathers give us long-term data. So it really depends what the question is, but we almost exclusively use hair in our analysis.
Okay, yeah, at least I would say hair makes much more sense than feces. All studies on feces, I know they have huge unexplained variation. So I have good experience with hormone levels being repeatable, but feces, I don't know what one is measuring there, but hair might be more interesting. Okay, then we have, but still one question, because to understand this, when you have on this epsilon axis, it's difficult to understand. But like when in your fetus cortisol, it goes from 0.5 to 1. This means the levels are double as high. Is that correct, or is that, or can we not interpret the data like this?
I showed the standardized levels because it was across different assays. But are you talking about the cortisol data?
Yeah, for example, that's what I have in front of me. I mean, but I mean, if on the epsilon axis it doubles from 0.5 to 1, it also means the individual has double as much hormone in the yeah. Okay. Yeah, but then yeah, like what you said, I mean, then cortisol there rather represents maybe metabolism, how much energy the fetus has available to grow and to develop, explaining why the ones with higher levels are more competitive, because it is not really something like a stress response.
Well, we know from human medicine that women that are in danger of giving preterm birth get dexamethasone injections, which is synthetic cortisol, and that allows the lungs to mature quicker. So cortisol is also a very important steroid in maturation of the different systems and preparing the fetuses for life outside the womb. And so we think that has to do with that as well, but also metabolically. I mean, all of these steroids, as behavior ecologists, we look at how they mediate the right reaction to the right context. But, you know, they all have a lot of different roles and different mechanisms and different systems.
Okay, thank you. Then we have a question by Rihanna.
Okay, do you hear me?
Yes.
Okay.
Yeah.
First of all, I'd like to thank Lee for this interesting presentation. I had many questions, but the thing is that I think that all the part of them was answered by your answer to Kirsten. And I have one particular question, because I'm not really familiar to the topics. Is there some phenotypic difference between female with high testosterone compared to female with low testosterone? With what the male will choose that I will mate with that females compared to mate with this one? Yeah, I think it's my only question. Thank you.
That's a wonderful question. For us, it's not detectable, but this is just for us. We know that for many steroids, there's smells associated with them. For humans, for example, we know that women, when they're in their follicular stage, prefer t-shirts of sweaty men that have high testosterone. And then later on, when they're in the luteal stage, they prefer sweaty t-shirts of men with low testosterone. So we know that we could detect testosterone levels by smell. We still don't know how to collect odor from hyraxes. We're now working with a researcher that is an expert in odors to try to figure out how to do that, because we're missing so much of the picture. I mean, we could only quantify what we see, and we don't see differences. We don't even know how to recognize individual hyraxes unless they have their colors. I mean, some of them are really obvious, like they have a broken tooth or a floppy ear, but a lot of them are just really, really hard. Like they don't have the spots of the spotted hyenas. And I mean, they're really hard to tell apart. So we can't really tell like who looks good and who's not for the hyrax, but they seem to know. So it's either through vocalizations, through odors, through behavior.
Or yeah, maybe the aggressivity as well. Maybe the male will not mate with a female who are really aggressive compared to the others.
Right.
Okay. Thank you.
Yeah, it's probably very complex. Like when you see somebody, you know, you have an opinion about them, and that sort of schemes, you know, it sums up so many different things that are hard to break up into and nominize them. So for an animal that's not us, it's even more difficult, but it's very interesting for me to try to find it out.
Okay, I think I forget to present myself. I'm Rina Ramanatsalama from the Deutsches Primaterium Centrum from the German Primate Center. Okay, thank you again.
Okay, then we have a question by Lindelani.
Yeah. Hi, everyone. My name is Lindrani Makuya, and I'm a PhD student working on the costs and benefits of solitary living in the bush corridor in the University of the Witwatersrand in South Africa. So my question is a bit of a continuation from the topic that everyone has been asking, because you mentioned that males have a higher testosterone level and this could lead to a higher success. So I wanted to know, is there any correlation to that with their ability to sing or how well they sing? And have you seen any difference in that?
Great question. So yeah, I mean, we've just published last year a paper that males that sing more tend to have more offspring. And we know that males that have higher testosterone tend to sing in a very particular way. There's an element called snort that only males that are dominant and have high testosterone use that sound. And that, I think, gives cues to the other individuals around as to their status, social status, and so on. We still haven't linked higher testosterone with more offspring, but since we do know that males that have higher testosterone sing better, and the males that sing better have more offspring, then I think eventually we'll be able to link that as well. But as we said, like that might take a while to have that direct link, but we're on our way yet there.
So I will see if there will be another question. There was none at the moment. So I would like to remind everyone next week we'll have a very interesting panel discussion on statistics and how editors of different journals relate to the importance of statistics, statistical analysis, what's acceptable, what is necessary. And I thought there's one more message, before I'm going to end. That was some time to think about. Eduardo has a second question.
Yes, of course, testosterone being such a prominent aspect of the talk, I have my brain getting around lots of doubts and questions, but I'm not, like some others have said, I'm not an expert, I'm not an endocrinologist, but I have to admit that it leaves me thinking a lot. I mean, this reliance on hair almost exclusively. So I have two questions or comments. Choose which one. One is, have you done ACTH challenges? Have you tested with very robust study design if the hair shows what you expect by challenging the animals? And this can be done if you're saying that hair is going to pick up different time scales, and we can tell them. I imagine it could be designed to test that in the lab, see if it really reflects what you've done. And the other one is that, going back to Kaya's warming us up to next week, is something that our young colleagues organizing the panel for next week, one of the things that came up from their discussion of issues that they worry about is power analysis. And I'm not going to say estimates of effect size, because it's really more like guesstimates of effect size. How are we going to handle in all of this? What does it mean? I mean, yes, we can do standardized testosterone values, but unless we develop the research methodology to actually test the responses of animals for various differences in testosterone, how are we going to evaluate what does it mean to have a difference of whatever magnitude we choose?
Yeah, good points. As to the ACTH challenge, there's another system that I didn't talk about, the Gerbils nanos, the Baluchistan gerbils in the desert. And on them, we've been doing GnRH challenges and ACTH challenge and blood testing and hair testing. We have not been able to pick up a single ACTH challenge in hair, but that's expected. I mean, after getting the ACTH shots, it takes about half an hour to see the peak in cortisol, corticosterone, and that goes away half an hour later. Whereas the hair is evaluating about a month of integration. So a single half an hour increase in corticosterone is not reflected in the hair. And I think what the hair does reflect is long-term different study sites, systems, different pressures. We could see, for example, that those that live beside agricultural areas have much higher corticosterone levels than those living in natural areas. So that's a long-term pressure that has to do with predator presence, also higher reproduction because of different, there's more resources, so they have more reproduction. Higher reproduction also means higher glucocorticoid levels, and so on and so on. So equating hormones with like stress with cortisol, I think is very, very challenging. I mean, I don't think of cortisol or corticosterone as a stress hormone. I think of it as a hormone as, wait, something's happening. Like we have to figure out what's happening. And we see glucocorticoids increasing almost at anything. And similarly, testosterone, it's a very reactive steroid. And the only way I think to really put it in context and looking at it long-term. If we had a system that we could take samples at the same second without handling the animals, possibly that could have been worthwhile. But if we're talking about wildlife that live in a natural environment, in natural context, I think hair is the best solution. Taking it a step further, you're talking about the power analysis. We always show estimates and so on, but the more and more I get into hormones, the more I realize that everything that we've been talking about, reporting steroid levels, is really very limited if we don't also report on receptor levels. Because it's not enough to just have the hormones levels. If there aren't the means to detect the hormone levels and carry it out into behavior, then it's really meaningless. And in the hierarchy, we're only starting out now. I'm sort of in conflict about it because I'm a wildlife ecologist and I don't want to kill animals. I mean, with a nutria, we have an advantage because they're being killed anyways. But in the hierarchy, we could measure testosterone. I mean, what Wingfield and the crew did was observe song sparrows behave, and then a minute later, they decapitated them, took them, and then were able to measure blood, circulating hormones, and the brain receptor hormones, which is beautiful. I love reading these studies, and I learn so much from these studies. But honestly, I cannot do these studies. First of all, I'm interested in long-term research. I'm interested in the well-being of these animals. They live for 12 years. I'm not going to observe behavior and then kill them and take their brains. So I'm sort of trying to get a proxy and try to understand it. And that's why I'm not just hanging on to a single observation. We're watching them long-term. We're watching them over the years. We're accumulating data. When we have an opportunity, and that's another advantage of long-term studies, we've now have quite a few bodies in the freezer. When we do see an animal getting predated on or killed, then we collect them. And now we're starting to look at the brains of the hierarchies as well and look at the receptor levels of males and females. Also, there are quite a few settlements in Israel now that have an issue with hierarchies overpopulating as well. And they're becoming an invasive species in Israel. And again, we're very politically active to stop the culling. And I mean, they haven't started yet to not allow the culling of hierarchies in certain settlements, but in other settlements where they're already okayed culling of the hierarchies, we had to put it in place that every hunter that kills the hierarchs must bring them to our lab immediately. And we already have a freezer full of hierarchies that were culled, not in our study site, but outside of settlements. And eventually we'll be able to say something about it too. But we have a lot of different things that prevent us from really investigating things thoroughly. I mean, if I was a different person working on a different system, I think I'd have much more insights and be able to say smarter things. But within my framework and what I'm willing to do, I'm trying to make ends meet and sort of learn the story and investigate things in depth and try to make sense of what I'm seeing of how they're behaving.
I just want to share with you. You didn't attend last week, but your presentation and your sharing of the ethical issues of the research, in my view, enriches so much. A conversation we had about ethics last week. I mean, one of the speaker was telling us how a study had to be stopped because the ethics committee, and this was in Finland, right? The ethics committee would not approve of them letting some cannibalism go on, cannibalism that is part of the natural repertoire of the animals. And so suddenly we're hearing about that from Finland, and we had this wonderful conversation. And really, for me, it speaks of the need to have a very broad view across countries, cultures, taxa, that comes from attending FINE every week. And now here we're hearing that, okay, if it's the animal well-being until it bothers humans, because suddenly how are we going to decide why is a Myocastor coipus as an individual, because what you kill is an individual, less valuable than a hyrax, right? And so they're okay killing coipus in Israel, but they're not okay on letting a cannibalism occur among frogs, tadpoles, or whatever. So it's fascinating. It's fascinating, and yeah. Thank you.
And I work in a nature reserve, and now there's a law that you're not allowed to kill cats, feral cats. So I see feral cats inside the reserve eating native rodents, while studies from 20 years ago in Israel that killed cats and looked at their stomach, at their what they ate, showed that they are, such amazing hunters and the found species that we thought were extinct in Israel, that the feral cats were able to catch and eat. So, I mean, ethics goes a long way, and what you are allowed to kill or not is very cultural, I think, also, and definitely worth discussing.
It's very challenging for us because I am assuming most of us in the sciences tend to have a rather rational brain, but more and more, I am getting old. The illusion that most social decisions are going to be made based on science, we'd rather wake up to it. We can do all the science we want, but the decisions, call it ethics. And I have a hard time accepting it. But there's also, I don't think we're going to change the landscape much. It's going to be whatever other sources of value, judgment, say, how to handle animal research. Thank you. Very interesting.
Okay, then we have a question by Sulema and by Raf. I don't know whether Raf it relates directly to what was discussed now, or whether we go first, Sulema.
Well, actually, let Raghavendra ask his question first, because I've already asked questions, and then come back to me afterwards.
Okay.
Can you hear me?
Yes.
Thank you. That was, I know very little about this, but that was absolutely fascinating. I'm becoming interested in this because now there's more and more interest in looking at similar things in insects, which was not there a long time ago. So I'm trying to learn as much of this as possible. My curiosity was, if there is such a strong effect of the fetus neighbor, the neighbor of the fetus, the sex of the neighbor, and so on, is there evidence or expectation that this will have long-term effects when these fetuses become adults and their ability to invest in parental care? I can imagine all kinds of runaway things happening. Everything, much of what adult animal does depends on who your neighbor was when you are in uterus. Is there evidence or expectation that this will have?
Yes, definitely. There's a lot of evidence, especially from model species. In mice, I gave one example that females that are in utero between two males are known to have higher testosterone as adults, be less fertile as an adult, and also be chosen. They're not less fertile per se. They could get pregnant, but their periods are longer, so they have less cycles. And the males prefer to mate with females that were not next to males in utero. So there is a way, again, maybe through sense of smelling higher testosterone, but males prefer females that were next to females in utero and not next to males. And also males that were next to males in utero tend to have bigger territories, be more aggressive. And yeah, there are many studies in rodents that show that this persists into adulthood. What you experience in utero has such far-reaching effects that it makes you wonder what we experience in utero and how that sort of shaped us into who we are today. And,
sure. Fascinating. Thank you.
Thanks.
Okay, then Sulema.
Yeah, and I guess I'm going to pick up right where Raghavendra left off. And I'm wondering whether in the hyraxes you do find any effects of intrauterine position of females other than the anogenital distance, if there are any behavioral effects or anything else you've been able to pick up for the females that are that have greater exposure to testosterone in utero. And then I have a couple of others, but I'll let you answer that one first.
So in the hyrax, we've only recently started doing similar things that we did in the nutria, because only now they're starting to be called in different cells. We don't have enough of a sample size, and also in Hyrax, as I said, everything is very strange about the species, and they don't have the uterine horns are not very clear. Like when you open up the Hyrax, it looks like a mess. It just looks like a huge tangled mess, but we're not actually able to fold it out. And I'm not even sure if it's relevant, because in the nutria, it's like a textbook animal. You open it up and the two uterine horns are spread out, and it's very clear who's next to who. So in the Hyrax, even if we'll be able to figure out if it's supposed to have two uterine horns, but they're all tangled up. And we're thinking that even if we'll be able to unfold it and know who's next to who, if it matters, because if they're all tangled up, they're all sort of next to each other. So they're all affecting each other. And we don't know if the steroids travel through the amnio fluids, or are they traveling through the blood flow? I mean, in the nutria, we were able to collect amnio and blood and everything. We're able to see the whole picture. But in the Hyrax, they're all touching each other and they're all interconnected, and it would be very hard to tease out and know. We are hoping to, I mean, once we have a big enough sample size, we'll be able to look at the sex ratio and see if there's a relationship between sex ratio and higher testosterone in females and so on. But we're not there yet. On one hand, we're waiting for more bodies. On the other hand, we don't want more to be called. So eventually we might be able to answer that. And in the field, of course, we don't know. I mean, once you have the fetuses are born, you don't really know where they were in utero. So
yeah, and I assume in nutria you haven't gotten to that point yet to be able to look for behavioral. Oh, they're dead, so you can't.
They're dead. Yeah, I had.
Okay.
Yeah. We tried marking some nutria and then following them, but it brought a whole slew, if we're talking about ethical issues about that. Then we had to tell the hunters not to kill our nutria, and it became too complicated, and we decided not to be involved because we're active against the culling. So then why are we telling them who to cull and not? So yeah, we can't do behavior on the nutria. We do observe their behavior, but they're not, since they're not marked. And both Hyrax and nutria, the males have abdominal testes. So it's not, we're not able to tell who's male and who's female just by looking. I mean, all other mammals, you see the testicles, you know who's a male. And both of these species, we don't know who the males are. So unless they were marked, we can't really, I mean, we observe their behavior, but we need the sex and the individual identity to be able to look at the context.
And then my second question has to do with testosterone being aromatized to estradiol. I wonder whether you have any data in either males or females that any behavioral effects are actually being mediated through estradiol after testosterone is aromatized.
Yeah, well, testosterone, we haven't been able to measure estradiol in hair, but estradiol is usually circulating at three orders of magnitude less than testosterone. We measured estradiol in picograms, whereas we measure testosterone in nanograms per ml. And that's because most estradiol is aromatized near the target organ. So it's usually aromatized in the brain next to the estrogen receptors, because they're highly mutagenic and carcinogenic, et cetera, et cetera. So in the brains of the nutria, we're able to, in the fetal brains, we do measure steroids. We were not done with that yet. We don't have data for that. So far, we've only been working on the receptors, but we will be doing that as well, and we'll be able to talk about the different steroid levels in the brains as well.
Great. And then my last question was whether in the Hyraxes that are mating with multiple males, do you see any evidence of higher reproductive success if a female mates with multiple males versus with only one male?
That's a great question. We only recently now finished the reproductive success. It was something that was supposed to be for my master's and for my PhD. And 20 years later, we only now have the data. They're very inbred. So using microsatellites and all other methodology was not effective. We eventually, once the Hyrax genome it's still not annotated, but once it was published, we're able to just sequence the Hyraxes and use SNPs. So we have just started to look at that, and we haven't been able, I mean, we've just done the first paper, the first study looking at male singing and reproductive success. Next up is all the testosterone data and asking questions about whether females that have higher testosterone and males having high testosterone have more reproductive success, and then starting to cross the behavioral studies with reproductive success. But it's in the pipeline. We're looking for great students. If you know anybody who'll be able to do it, we're really searching, and we'd love your help to send people our way. But we have so many questions and so much data, and we're just slowly going through it.
Thanks, Lee.
Okay.
Then we have a question by Francine.
Thank you. Thanks for a really interesting talk. It's fascinating. And also the discussion about ethics is really important as well. So thank you for that. My question is, does the location where the female fetuses are in utero impact their mothering quality or ability? So if they were surrounded by males, are they more aggressive mothers or less aggressive mothers? Are they better mothers? I'm just interested in, I think that would be a really fascinating question. And then what impact does that have on their offspring? Are they more careful? Are they, better? Is their survival better? Are they better at finding food? I mean, like there's all sorts of questions related to that. But anyway, thanks so much.
Yeah. Great questions. So again, in the nutria where we do have the intrauterine possession, we don't have any behavior because they're dead. But from the literature, from model species, from mice, it has been shown that females that are in utero next to females are better mothers. They have more male biased litters. They also have more fetuses, so they have bigger litters and higher reproductive success. So
these are females who have more males near them in utero?
No, these are females that have more females near them.
More females. Okay, thanks.
Yeah, so at least for mice and rats, the evidence has been very consistent across the board. So being next to females are better off being next to females in utero, and being next to males masculinizes them behaviorally and physiologically and reproductively, and that carries on into adulthood and is detrimental for their success. But again, in the nutria, things are really different. We see different results, and we don't have the behavior, unfortunately. So we can't ask these questions. In the Hyrax, we looked at females that have higher testosterone. We wanted to see if they're worse mothers than females that have lower testosterone. And we found that females, that testosterone was not related to how much females nursed and how attentive they seemed to their offspring. But we cannot quantify how much milk was given, like just quantifying the time of nursing doesn't necessarily quantify the quality of nursing. We don't know how to do it. Like we're constantly observing and sort of tuning in and thinking, what else can we quantify? How can we do things to be sort of more informative? But it's a question that I'm really interested in about mothering styles and how testosterone could be affecting mothering. And again, with the right student and the right observations, I think there's a lot of interesting things to find in that way.
Thanks so much.
Okay, yes, thanks again, Lee, for a very great presentation, and was very interesting for all of us to hear about these long-term studies over 25 years now and how this all develops. Thanks to everyone for your contribution to a lively discussion. I think we all learned a lot today again, and looking very much forward to see you again in 1 week when we will have the discussion with the editors of the different journals. One editor is here, Raf, and then we talk about how to deal with statistical issues and problems and so on in publishing.